Hello, my name is Mark Essien, and my project is entitled The Loss of E-cadherin Alters Epithelial Plasticity in Normal Human Bronchial Epithelial Cells. So first, I'll begin with a little bit of a background. COPD stands for chronic obstructive pulmonary disease. It is an inflammatory lung disease that causes obstructed airflow in the lungs. It is characterized by chronic bronchitis and emphysema. Symptoms of COPD include cough, mucus production, and difficulty breathing. COPD mainly occurs in individuals who smoke. However, it is becoming more common in areas where air quality is poor. To better understand COPD, we need to discuss how COPD occurs on a cellular level. Our epithelial cells are the first line of defense versus stimulants. These cells make up our epithelial barrier. The epithelial barrier is maintained by adherence junction proteins such as E-cadherin that keep the cells tightly packed together. After long-term cigarette smoke exposure, E-cadherin expression levels decrease and this barrier breaks down. Cilia height decreases and cells undergo metaplasia. This is detailed in the diagram here on the right, where the epithelial barrier is shown along with the breakdown of the epithelial barrier, as we can see with this leaky barrier here. In our study, we will look more at the role of E-cadherin and whether it has a causal relationship with COPD. In my project, I hypothesized that E-cadherin knockdown is sufficient to cause epithelial plasticity, similar to what is seen in COPD patients. Our objectives were to knock down E-cadherin in non-diseased human bronchial epithelial cells and determine how E-cadherin expression affects epithelial plasticity. We did this by culturing normal human bronchial epithelial cells at air-liquid interface and performing an E-cadherin knockdown. To confirm that the knockdown occurred, we analyzed our cells through a Western blot. Next, we analyzed tight junction integrity through transepithelial electrical resistance, or TEER test, cell permeability through Fitzy-Dextran, cell velocity, and finally ciliary beat frequency to determine how fast cilia beat. CDH1 is the gene that regulates E-cadherin expression. SH-CDH1 is our experimental group, while GFP is the control group. As we can see in our Western blot and E-cadherin expression chart, the CDH1 expression level has a significant decrease in E-cadherin expression level, as we can see here. The SH-CDH1 group had a significant decrease in TEER test results, as seen in this graph here. It also had a significant increase in cell velocity, as seen in this figure. However, the CBF and Fitzy-Dextran tests had no significant difference between the two groups, as seen in this figure and this figure. In conclusion, we achieved our E-cadherin knockdown and discovered that this led to significant differences in TEER and cell velocity. But there were no significant differences in Fitzy-Dextran permeability and CBF. We hypothesized that this ambiguity exists in the results because E-cadherin plays a bigger role in monolayer formation rather than monolayer maintenance. We also hypothesized that E-cadherin regulates tight junction function. In future studies, we would like to overexpress E-cadherin in COPD cells. That is all for me. Thank you. I would like to thank my mentor, Dr. Sadeh. I would also like to thank Bashaki, Bonnie, and all the members of my lab who have helped me this summer. Thank you.